Okay, what is up, my Form 4? It's a Monday video. Okay, this video we are going to talk about exercise and might talk about a bit about cloning if you have the time. Okay, I hope you understand the previous video. If you don't, please watch it a few more times. Get it under your belt. Okay, make sure you understand it fully. Okay, all right, let's get into the question. Remember we drew the previous lesson. Okay, so today let's revise a little bit what's happening here. So the first phase, okay, we are in mitosis. Okay, this is called prophase. Okay, do you still remember what happens in prophase? Okay, so you can say chromosome, okay, visible under light microscope. Okay, and what happens to the surrounding? Okay, the nuclear envelope, okay, disintegrate. Okay, disintegrate means it disappears. All right, so second phase, the one that I said they like to ask the most, this is metaphase. All right, metaphase. So what happens here? Chromosome, okay, chromosomes, okay, whatever, okay, arranged at equator. All right, equator. Remember the line is called equator or metaphase plate if you like it. All right, the spindle fiber. Remember the spindle fiber? Spindle fiber is the thread, okay, the string, okay, attached at centromere. Okay, centromere is the middle dot, remember? All right, and then the third phase, we call it anaphase. Okay, pasar malam ada telephone, remember? That's my way of pro maths, the way you remember. Okay, so anaphase, what happens here? Sister chromatid. Okay, separate. Remember what is sister chromatid? Chromatid, sister chromatid, chromosome. All right, sister chromatid separate. Okay, move towards opposite pole. Okay, opposite pole. You have north pole and south pole. Okay, whatever, you don't need to say this. All right, the last phase, we call it telophase. Okay, telophase. What happens here? Sister chromatid reaches opposite pole. All right, reaches opposite pole. Nuclear envelope, okay, develop, or you can even say chromosome, okay, it becomes invisible again. All right, so as I said, 2 points for every phase. You don't need to, full-blown elaboration. Okay, there's no need. All right, easy way to score. All right, let's look at this question. Cell cycle. Okay, name the label at the diagram. So what is C? Okay, let's not start with C, sorry. Let's start with X. What is X called? X is my G1 phase. Okay, G1 phase. Y is my S phase. Z is my G2 phase. All right, M is the mitotic phase, okay, or mitosis. Okay, C is my cytokinesis. Okay, cytokinesis, the separation of the whole cell. All right, so draw an arrow to show the interface. Okay, so interface is here. Okay, the whole thing is interface. This tree is interface. All right, so 1 2 3 4, you have P M A T here lah. Okay, so C, for each phase of the interface stage is main cellular activity. All right, so G1, what happens here? This is called the growth phase. Okay, so growth, okay, or synthesis of organelles. Remember, before I separate my cell, I need to have 2 sets of every organelles. I can't split it into half. Okay, so S phase is to synthesize DNA. Okay, it's to photostat the DNA because I need to split it into half. All right, G2 is further growth of cell. All right, very simple. Refer it to the notes. This question is not common. Okay, I wouldn't advise you to focus. All right, D. Describe how the arrangement of chromosome during M2 differs from arrangement of chromosome during M3. So M2 is metaphase. Okay, M3 is anaphase. So arrangement of chromosome differ. Okay, so at metaphase, don't say metaphase, okay, it says M2, so let's stick back to M2. At M2, it is arranged at equator. Okay, at M3, they are separated. Okay, or you can say move towards opposite pole. All right, simple. E. Process C is different from animal cell. Explain differences. Okay, remember. So for animal cell, you have something called cleavage furrow. Okay, so what is cleavage? Okay, cleavage. Okay, I don't have it. Okay, my chest is not that big. Okay, so plant cell, this is cell plate. Okay, so refer to the notes, you get it. Okay, F. State one difference between karyokinesis and cytokinesis. Okay, what is karyokinesis? This is separation, okay, of nucleus. Okay, this is not a very common question. Cytokinesis is separation of cell. Okay, so cyto, okay, cytoplasm, I'm separating cytoplasm. Karyokinesis, karyo actually means DNA. Okay, so I'm separating my DNA, which is the chromosome lah. Okay, DNA forms chromosome. Okay, chromosome is a whole bag of DNA. Okay, if it makes sense. All right, let's look at question. This is a common question. Okay, just now the previous question is not very common. This is very common. Label R, S, and Q. So what is R? R is my nuclear envelope. Okay, nuclear envelope or nuclear membrane. Okay, so S is my centriole. Okay, Q is my chromosome. All right, simple. So what is the function of Q? What is the function of a chromosome? This is a carry genetic material. Okay, so simple. So name the stage in figure 3. So they are all hanging around. What is this? This is prophase. Okay, like I said, a whole bunch of hair. Okay, of course, this is not that long. So number 2, describe the chromosome behavior, name in B. So chromosome. Okay, what is it? Visible. Okay, under light microscope. Okay, light microscope, not electron microscope. Electron microscope, you can see everything. Okay, so number 1, C1, name the following stage after stage shown. All right, so simple. Metaphase. Explain. Okay, the keyword here is equator. Okay, chromosome arranged at equator. Okay, write your full answer. D, what is the number of chromosome in the daughter cell produced? Yes, okay, this is a very important question. Okay, please highlight this. Okay, so can you see how many here? 1, 2, 3, 4, 5, 6, 7, 8. Okay, 8 cell. 8 chromosome. When I divide into 2 cells, I would have 8 and 8. Okay, because mitosis is to create the exact same cell. Okay, you won't change color over time. Okay, unless you're an iguana or whatever. You won't change appearance over time unless you go for plastic surgery, because all the cells created by mitosis is exactly the same. Okay, all right. Some will count like this. Oh, sir, sister chromatid. I count this as 1, 2. No, this is still one chromosome. After I separate them out, this is one chromosome. After I separate them out, it looks like this. This is still 1, 1. Same number of chromosome. Can you see? Okay, I can't create different chromosome out, then I would look different. All right, so 8 chromosomes here. So number of chromosome in daughter cell, also 8 chromosome. Okay, they just want the number, just say 8. All right, a lot of people get this wrong, so please don't. Okay, a very simple question. All right, state the importance of this cell division. What is mitosis for? Okay, to repair or replace, okay, dead cells. Okay, growth. What else? What is that thing called? It's a asexual reproduction. Okay, so many, just one would do. All right, let's look at this question. I would say this is the same. Okay, oh, there's a drawing one. Okay, you draw it yourself. This is prophase. So what is the following phase? Metaphase. How do you draw metaphase? Okay, very simple. Centriole. Okay, you have your spindle fiber. Okay, here's the question. Can you see how many chromosomes here? You have 4. So make sure you draw 4. I hate to say it, some teachers, they are very fussy about this. You can get wrong. Okay, so don't make mistakes like that. All right. All right, let's skip ahead. Yes, question 3. I want to talk about cloning today. Okay, we didn't talk about cloning because the video was getting a bit too long. Okay, so basically what is cloning? Cloning is basically to create the exact same organism. If I were to clone you, I would make another you. Can you imagine how scary is that? Okay, I can clone an army of myself. Okay, so oops. All right, sorry for that. Some problem with the iPad. Okay, let's talk about this. Okay, yeah, cloning. So what is cloning? Cloning is to create an exact copy of yourself. Okay, can you imagine an exact copy of yourself? We have not done this onto humans. Okay, the furthest we have gone is a sheep. Okay, a sheep in Edinburgh, Scotland, actually. Okay, this experiment was done in 1990 something, I couldn't remember. Okay, we used to clone, we still now clone vegetables. Okay, to move this into animal territory is unheard of, and it's very ethical as well. A lot of people say, oh, who are we humans to play God? Okay, how can we clone animals? Okay, a lot of issues arise from this. So we have not done any cloning since then. Will we do it in the future? I don't know. Okay, it will be like Star Wars already. I will have a clone army, okay, fighting each other. Okay, very controversial issue. Okay, just like gay marriage. Okay, I'm not gonna talk about that. All right. So what happened is, what happened was when they did this experiment, it failed a lot of times. Okay, they failed a lot of times. Basically, what the concept is, okay, now imagine I have 2 sheep. Okay, so I take, this is a female. Okay, this is a female sheep. I take the ovum out. Okay, the ovum out. Your ovum has 1 set of chromosomes. Remember, you have N. We are 2N. Okay, we are 2N. So the ovum is N. Okay, 1 set. We are 2 sets. So this ovum is to combine. We'll talk about this in meiosis. This ovum will combine with the other end of the sperm. Okay, to form you. You are 2N. So now I've half, which is 1 set. So what they did is they use electric and then they remove the nucleus. They throw away the N. They don't want the N. Okay, and then from this another donor U, they take a cell. Okay, any somatic cell. Okay, remember it's somatic cell. Somatic cell is any cell in your body. Okay, any somatic cell. So they take out the somatic cell and then they extract the 2N from the somatic cell. Means let's say I take one of your skin cell, I take the DNA. So the DNA is from you have 2 sets of DNA, one's from Papa, one is from Mama. So I straight away take that. And then they use electric, the electric actually should be here, they zap them together. Okay, so this immediately forms an embryo. Very funny, very scary. No sperm and ovum here, because the ovum already throw the DNA away. Straight away, this is you. I grow another you. Okay, I grow another you to make another you. And that is pretty scary if you ask me. Okay, yeah, and then they take this and then they put it into another surrogate. Okay, you know a surrogate? Surrogate means I borrow your uterus to have baby. So they put another surrogate sheep. Okay, they put it inside. Then in few months, I create another exact sheep like donor U. Okay, because I'm using his cell, let's assume this is a human. I'm using his cell. Of course, I'm creating another him. If it makes sense. Okay, I take one of a skin cell, I put it in an ovum without anything, and then I grow another you. Okay, isn't that scary? So, they did this a lot of times. It failed a lot of times, and then they successfully did it once. Okay, and it created an uproar throughout society. Okay, so they said this is very unfair or so ever. Okay, playing God, whatever. Okay, so funny story is, and oh yeah, the sheep, they named it Dolly. Okay, go and search for Dolly sheep. Let me show you. Okay, this is the Dolly sheep experiment. So yeah, basically this is it. Can you see? Okay, an egg cell. Okay, we are looking at the left side. So there's an egg cell, and then I remove the nucleus. Can you see? I remove the nucleus, and then from the donor cell, okay, I take out one nucleus, and then I use electric zap into it, and then I put it into an surrogate. Means I borrow the uterus to pregnant, and then I produce Dolly. All right. So this, unfortunately, Dolly didn't live very long, only for a few years. Why? Because the original mother, okay, when they cloned her, she was 9. I think she died at around 14. Okay, so how many years there? 5 years. So Dolly died at 5. Can you see? Why? Because once they clone Dolly, at the first year, she's already 9 years old. Because I'm taking the 9-year-old cell, I clone a bunch of 9-year-old cells. Of course, the sheep is going to be 9-year-old. Okay, 9 years old by the time when she was born. Pretty scary if you ask me. Can you imagine seeing another version of yourself? Okay, so there you go. That's cloning. Okay, do we clone anything now? Anything? Yeah, we do. Okay, actually most of your bananas are cloned. Okay, it's not from pairing. Okay, from breeding. It's from cloning. All right, so let's look at the question. So define cloning. Okay, cloning is to create, okay, another identical organism, okay, via mitosis. Okay, the keyword is identical, exactly the same. There's no variation. Imagine I were to clone 1,000 of me, 1,000 of me will look the same. Okay, you will have 1,000 Eddies. Okay, so anyway, number 2, name the species of sheep that will be reproduced at the end of the experiment. So who am I producing? I'm producing donor you. Okay, because I'm using his cell, her cell, okay, whatever. State 1, disadvantage and advantage. So what is the advantage? Okay, quick reproduction. This is very fast. Okay, like I said, in 1 year you can create an adult sheep. Okay, what else? It is identical. Okay, you might say, so how is this good? It can be bad or good as well. If I have a lot of good genes, I create a few more thousands of me that has the same good gene. Okay, so disadvantage, this is also the disadvantage. Okay, there is no genetic variation. Okay, do you know this helps us to survive? Okay, how so? Okay, imagine now I have 2 types of birds, A and B. Okay, A and B. Now there's a disease. This disease kills A. It kills A. Okay, A is very vulnerable to disease. Somehow B is immune. Okay, because of this variation, A will die out, but B, I will keep on reproducing. Okay, law of nature. It is because of this variation, this species, okay, although they are different because they are variation, because of this variation, this species is able to survive. Okay, it's able to create offspring, which is like us. Okay, we adapt to our environment. If you look at people on equator, for example, they have lived on the equator for a few hundred years. Their skin is really adapted to the UV. Okay, that's why they look dark. Okay, our Malay friends, they look dark. Our Indian friends, okay, they are from India. India is on the equator. They are dark. Brazilians, they are dark. Africans, they are dark because of the sun. Okay, so can you imagine last time there's no houses whatsoever, we are cavemans and all, so we are exposed to a lot of sun. So they developed the protection against sunlight, whereas okay, as me or us, we are Chinese, okay, we live in we came from China, okay, it's only been like what, 3 months of summer, there's not much sun, we evolved into not having so much dark pigments in our skin, okay, because dark pigment absorbs a lot of heat, okay, it's not very necessary, that's why we evolved into becoming fair skin. Okay, so if you go upper some more, which is the Nordics, okay, Sweden, whatever, they even lost the dark pigment in their hair and their brows. That's why they are blondes. Okay, in their eyes as well, they lost the dark pigment. That's why they have blue eyes. Okay, can you see we adapt to our environment differently? That's why this is what makes us survive. All right, because of the genetic variation. If all of us were to look the same, okay, so imagine just now this species, okay, now I only have species A, I only have type A. If a new virus comes that kills type A, gone case, all of us will die. Okay, this gene, this species would cease to exist, it will be extinct. Okay, I hope this makes sense. That's why we need the variation. All right, so yeah, like we talked about this just now. Banana is the most important fruit crops in India, the largest banana producer in the world. They employ commercial tissue culture. Okay, what is tissue culture? Tissue culture is basically cloning for plants. Okay, plants cloning. We call it tissue culture. Okay, basically the same concept. We take a piece of the tissue, we just photostat them out. Okay, so technique for mass production of popular banana variety as shown. Name the method used. Okay, I'm not sure what they want here. This is tissue culture. Okay, tissue culture. You can't say cloning here because cloning is for animals. Okay, so I think they gave the answer here. All right, so B, what is the characteristic? Okay, they are all genetically identical. Please don't say same. Same is wrong. What same? Okay, genetically identical. Name the solution that the tissue are immersed and what it contains of. Okay, it's in the note actually. Okay, let me point you to, yes. Okay, so they are, I think it's actually called culture solution. Okay, it's not mentioned here. Okay, it's called culture solution. Okay, what does it contain? It contains nutrients. Okay, obviously because it needs to grow. Okay, two advantages. Okay, I already mentioned just now. Advantage and disadvantages. All right. So can the same method be used on animal cell? Okay. Okay, you might say no. Okay, because animal cells, okay, do not divide or do mitosis readily. Okay, that's why we need to put the cell into ovum, because ovum can divide readily. It will stimulate the DNA I put inside to divide actively. Okay, you can't force a healthy cell to keep doing mitosis unless it's a cancer cell, like I said. Okay, cancer cell will keep doing mitosis without ending. That's why you have tumor. All right. Okay, so that would be it. Okay, question 4 is almost the same. Again, cloning and tissue culture. Not going to talk about this. So this will be it. Okay, thank you for paying attention. Again, next week we shall go into meiosis. So please make sure you are ready. It will be a tougher subchapter. Okay, it utilizes the infos you have now. That's why please watch mitosis a few more times, because it will be slightly more confusing in meiosis. Okay, we will do it twice and there's a lot more things to draw. All right, I hope this is helpful. I hope you are paying attention and I hope you understand. All right, that's it for me today. Thank you for paying attention. Thank you guys, and I will see you soon. Bye bye.